welcome to our hazardous waste, or at least the waste chapter virtual lab. With this one, you're going to fill out a typical design your own experiment. Of course, I'm going to run the experiment here, and you can fill out the paperwork there. Your question: well, what happens when a strong acid interacts with a strong base? If we're dealing with hazardous waste, it's not unusual for them to be a strong acid, strong base, something of this nature, and what happens when one runs into the other. In this case today, we are working with materials, hydrochloric acid, HCl, and we are working with sodium hydroxide, NaOH, sodium hydroxide. They both can be very strong acids, very strong bases. I have some aqueous solutions mixed up of each. Now these are not full strength, but they're still pretty strong. How strong are they? Let's take a look. So I want to switch into some gloves for this, so give me a second and I'll be right back. All right, now I have some proper hand protection and proper eye protection. I'm not too worried about vapors, but there is the possibility of something splashing, so I don't want it to get into my eyes. Well, let's take a look. Over here, this is the NaOH, sodium hydroxide. And we're going to add some into solution here. And over here I have my hydrochloric acid, or HCl. And we're going to add about the same amount there. Now let's kind of check. Now fortunately the hydrochloric acid is a bit of a color difference, making it easy. Now we are just going to see what happens if they are exposed to something as simple as tin foil. Now I'm going to get these kind of ready, just a small piece each. I'm going to drop them in there, and I'll try and bring them up to the camera, and we'll time lapse this to see what happens. So I just get these sort of equally crumpled up. Now, here we go, our two little pieces. I'm going to drop them in, I'll bring them up, we'll time lapse it and see what happens. So one in the sodium hydroxide, one in hydrochloric acid, and here we go. All right, so hydrochloric acid worked a little faster on the aluminum than did the sodium hydroxide, but the sodium hydroxide is still eating away and dissolving at the aluminum. We'll take a look at them at the end of the lab, but just to show you the effect of either of these. Either of these tossed out into a white waste site or dumping into the ground, etc., can obviously cause problems for human beings or other living organisms. So let's move on to the rest of it. What happens when a strong acid interacts with a strong base? Well, let's go back to our old friend, universal indicator. Now I've already got it mixed up, but universal indicator, we mix it in and we have a nice chart. Gives us our color. acids, hydrochloric acid on this end. Acids tend to show up as a red to bright pink. Neutral colors are in the center, so we have a neutral. And our blue towards purple for strong base. So I'm going to kind of flip this around so we can see the colors, and let's go ahead and add. So we're going to add equal amounts. And it might take me a minute to get these actually equal, but let's see how we're doing. Not too bad. Let's add a little bit here. That gives us pretty much equal amounts of our universal indicator. So let's see what's what. I want to try and do equal amounts. I'm going to try and go for 3 drops, but if I accidentally get 4 drops, we'll have to go 4 and 4. But let's see. So once again, sodium hydroxide. Do a few test squirts here. So 1, 2, 3. I got 3 pretty good there. I don't want to leave this in it long because it is kind of a strong enough solution. I just have a beaker of water down here that I can kind of rinse it out in. And if we shake that up, get a pretty bright purple color. And over here, where'd it go? My pipette. Same thing, I want to try and get 3. 1, 2, and 3. Same thing, I just want to rinse this one out real well so it doesn't potentially dissolve it up. Should be done with those pipettes, and yeah, wow, look at that. Really nice, bright pink. So, for you, when you're looking at it, you had your question, you need to come up with your own hypothesis about what would happen. Just so far we've seen what happens. And at this point, yep, the aluminum is completely gone. And the aluminum is completely gone. The sodium hydroxide definitely way out here on this dark purple end. And obviously pretty far out here on this bright pink to red end. So, as we would sort of suspect, we have a strong base. You can let me know what you think its pH would be based on our color chart. And we have our hydrochloric acid. Once again, let me know what you think its pH is based on the color chart, because we need that for our data, what we're trying to see. How strong of an acid is it? How strong of a base is it? So now we want to have them interact together. So just have a symbol container here, and we're going to mix them together and see what occurs. Now, we'll probably go a bit of both, but I may have to do a little bit of playing around to get the amounts just right. So let's see it. So I have added my sodium hydroxide. You should be able to see it reasonably against my coat. And if I add, still saying purple, took a little bit more, eventually did take it into, I'm going to go back to, I think I can use this one. And yeah, it didn't take much at all to take it all the way in, but we're more at a maybe a bluish hue now, if we can see it up against the background. So if we take them and mix one with the other, eventually we can take a strong acid and a strong base as another way of comparison and get back to a neutral. Now, it wasn't exactly, if you notice, half and half. If they had been exactly the same, we could have just dumped them in together. But in order to get it back to a neutral, took a little more nuance, I would say. At this point, you should have enough data. You can see what happens to aluminum foil in sodium hydroxide. We can see what happens to aluminum foil in hydrochloric acid. We could determine the pH of each, and ultimately what occurred when we mixed them together. Well, that pretty much wraps up the experiment. You now need to draw your conclusions. Well, was your hypothesis supported or not supported, or was your hypothesis inconclusive? It all depends on what your original hypothesis was. Now, what next? Obviously, we're trying to show what could happen if we're talking about a hazardous waste, if we mix this chemical with this. Of course, if it's an acid, a known acid, and a known base, we can neutralize them. Now I have something that is actually non-toxic that can be disposed of safely. If you can't think of something else, another experiment you would like to try, big scale. How might we try this on an industrial scale? Now, you've got 3 questions I want you to answer. You always need to identify your independent variable, the dependent variable, and the control. Ideally, we have an experimental group and a control group, so we want to identify them apart. First off, independent variable. I always ask myself this question: what did I change? What did I change? That is our independent variable. The independent variable is what changed the dependent variable. But I always ask myself this very simple question: what data did I record? Literally, what did I write down for data? That's my dependent variable, because that's the thing that changed. That's what I want to know. Lastly, the control. Sometimes it can be difficult to determine what the control is. Well, once again, I ask myself a simple question: what was the normal? What would we usually do? What was the normal in this whole setup? And that lets me know what was the control, what I was measuring my experimental group against. Well, that should do it. Hope you learned something, and wrap that up, get it to me, and we'll see you next time. Take care.